take this remote work, so it'll make things a lot easier. So, good morning, everyone. Good morning. So, this is a great class. Elena's doing a great job with it, and got good TAs, so you're in for a good, 3 months?
Yes, and because I come to every lecture, we try to come to most lectures, you feel it.
It's probably just here.
Okay, so we're going to talk about human genetic variation, or what follows here. And my 3 lectures will all sort of blend together. And you'll have a special treat Friday from a lecture from Eric Reed, who's head of NHGRI and has been head of NHGRI for some years now. And we're really fortunate that he makes the time in a very busy schedule to drive up here and give this lecture. So that'll be a special treat on Friday morning. Okay. So what is genetics? I'm trying to remember what we've discussed in that concept. So if I repeat myself, well, it's because I think it's important. I think genetics is an information science that concerns itself with the flow of information from the genome to the phenotype. And you can think of the phenotype, or I think of the phenotype as sort of a developmental phenotype, and then a homeostatic phenotype. Homeostatic is sort of the phenotype of the moment. Development occurs over the lifetime of the individual. Variation in this information, so it's that flow of information and then variation in the information, how this variation is transmitted across generations, and how it is distributed in populations. So we'll talk about various aspects of all of this. And the key point, of course, is that the genetic program interacts continuously with the environment and is constantly under natural selection. So a big difference over the time that I've been involved in genetics is this idea of the immediacy of evolution. I went to medical school, which was in the mid-60s. The word, I'm pretty sure the word evolution was never once mentioned in the 4 years I was never once. And at that time, evolution was sort of thought of as paleontology, what we might consider paleontology nowadays. And of course, if you think about it, we'll maybe have a discussion on some of the other lectures. Evolution is ever present, and it explains why some people get sick and why other people don't get sick. And so it's a very important idea and a very important topic for human genetics and for medicine. And so what's changed over the time that I've been doing it is it's gone from something that was never mentioned to now something that should be right in the front lines. And if you ask yourself, why is that possible? Well, the major reason it's possible is because of DNA sequencing. Because back when I was a medical student, evolution was basically paleontology. And now evolution has to do with analyzing the genome sequence, not only of our own extant species, but also our predecessors, and then across the biological kingdom. So it's really a great change, and it's really, for me at least, makes biology and medicine much more living, basically, active. Okay, so here's the information flow, and I may have shown you this slide before. If I haven't, I apologize. So there's the genome with the information, and that information is first transcribed and then translated into proteins. We know that the proteins don't work in isolation. Almost all proteins interact with other proteins. So in this ball and stick diagram, each ball is a protein, and then the connections, or the so-called edges, show the interactions of the various proteins, and they form a biological system. Ultimately, I think in certainly your lifetime, I think we'll have an alphabet of all biological systems. We're getting there now if you think about it, but I will have an alphabet of all biological systems and how each one of them works and what regulates them. That's something that you folks will put together. And then, of course, those systems conspire to make a unique individual. And the environment pushes against this pathway at every level. So it's a constant dance between your unique genome and your unique collection of environmental experiences. Okay? All right, appreciation of variation. And by here I mean variation in our genome and how it works. And if you, since I know that you all remember everything we learned in concepts, you could recite this slide for me, but just in case you're a little foggy on the details, we talked about Darwin in 1856 and his interest in and utilization of his observations on phenotypic variation. Garrett in 1902, biochemical individuality. Morgan in the fly room, 1910 to 1920, they used phenotypic variation to recognize rare variants in the phenotype and consider the wild type, that is, the flies without the particular phenotypic feature, as a monolith, that is all the same. We know that's not true now. Beadle and Tatum, one gene, one enzyme, so that we got into the idea that gene function involves proteins. And then we saw a lot of papers related to electrophoresis, methods to detect protein variations, so now we begin to develop technologies that expose that variation. And then Harris and Lewontin, Harris in humans and Lewontin in flies, exposed genetic variation as being extensive in both flies and humans, in fact, almost to the exact same extent. And that variation was a universal phenomenon in biology. And the Human Genome Project, 1990 to the present, DNA sequencing, genotyping, and variation galore. So really, over a relatively short period of time, we've gone from just saying, well, that pigeon doesn't look quite like the other one, so there must be some explanation for that. That's what Darwin got. That's what got Darwin started, and now we can explain it, and we know that what we see, the cursory glance, is really just a very tiny tip of the iceberg of the extent of genetic variation. In Darwin's time, they had produced all of these various pigeon variants, and many people thought they were all unique species. Darwin said no, they're probably all derived from the common rock dove, which is the common pigeon you still see outside here, through selection and breeding. And those observations, which he describes in the very first chapter of The Origin of Species, led him to help develop his ideas on evolution. So genetics deals with genetic variation, sources, distribution of populations, transmission, and phenotypic consequences. Okay? We're all in agreement, I think. Now, some definitions. These are just definitions that I use. They're not perhaps universally appreciated, but I find them useful. So genetics is the study of genes and genetic variation. We've just been talking about that. I'm going to repeat all that. So what about genomics? Well, from my point of view, the difference between genetics and genomics is that genomics study is the study of genomes, the entire set of genes. And so as genomics finds abnormalities, then we use genetic methods to study those individual abnormalities. So a human geneticist by the name of Peter Goodfellow pointed out that genetics is the parent science, and genomics informs genetics. The information we get from genomics then gives us the genetic variation that we use genetics to explain phenotypes. So the functional consequences of genetic variation. I guess, well, we won't turn on the light, but you've probably seen versions of this kind of slide before that. If you look around the room here, you can just say that, well, I'm pretty sure that everybody in the room represents a member of Homo sapiens. But everybody in the room is different. And I can recognize the differences. And So we're all human, but each of us is individually recognizable. And of course, one leap was, it was staring us right in the face, but one leap was that all of the, there's no reason to think that just as we have minor differences in our appearances on the externally, we have the same variation in internal things. And that, of course, is various contributions by some individuality. So we can imagine that if we could look at the Krebs cycle or glycolysis or whatever, we would say that's the Krebs cycle, but look, this guy's Krebs cycle is slightly different, and to the next person, that kind of stuff. So in a broad way, the way I think of genetic variants, of which there are many, and we'll talk about how many there are, but a large fraction of the variants are neutral. They don't seem to make any difference in sort of the selection on an individual. And another way to think about that, of course, is since we know that much of the only 1.8% or 1.5% of the genome is coding sequence, there's a large chunk that's regulatory sequence, but there's a large chunk that's competitive sequence and so forth. So a lot of that variation in those sequences doesn't seem to have any effect on the survival viability of the individual, so completely neutral. There's a smaller fraction of variation, which depends on who is present in and under what circumstances that individual finds themselves. So I can say smaller fraction depends on in who and under what circumstances. And those would be variants that doesn't seem to have any effect on me until I get COVID-19, and then all of a sudden that COVID-19 explodes this variant that's just sort of sitting in my genome, didn't seem to have much effect under normal circumstances, and now all of a sudden I'm very vulnerable to COVID-19 or to smallpox or to starvation, all the vicissitudes of environmental stresses that we deal with. And then a very small fraction of the variation is deleterious almost always. Okay, and those are rare variants for Mendelian disease. So you have a bunch of genetic variation, and its consequences vary considerably from anywhere from neutral to always pathologic. Now, one place you see that variation and the consequences of that variation, if you are an astute observer of biology, is in medicine, because in medicine we see individuals that are under stress, either because they've come across some environmental variable that they can't deal with, let's say an infectious agent or a toxin or some other environmental variable, and that makes them sick. And other people see the same environmental variable and don't get sick. So Osler, who was the chairman of medicine here many, many years ago in the early 1900s, said that variability is the law of life, and as no two faces are the same, so no two bodies are alike. And no two individuals react alike and behave alike under the abnormal conditions which as well as disease. So the way I think of this is that we have this variation, some of which is quite apparent under normal circumstances, but when you stress the system, let's say with an illness, that variation becomes all the more significant, easier to see, and of more consequence. So it's not surprising that a thoughtful physician would come to this conclusion. And of course, my mentor, Barton Child, said in biology, whatever it is variable. So these are two very thoughtful physicians. And they came to the same conclusion. Okay, so the central goal of human genetics is to connect this genetic variation to the phenotype. And that helps us to understand gene function at the cellular level, at the intact organismal level, understands human disease causation, how disease works, and often suggests treatment. So it's a really rich vein of information that we can mine to help us understand the organism in much greater detail. Okay, so that's a sort of an overview of genetic variation. One thing that I don't say in there, which I should have, was, well, I've sort of discussed variation all from the point of view of when it is harmful. And you could ask, well, wouldn't it be better to get rid of all that variation, to just pick a program that had no harmful variation in it? And why can't we do that? Why doesn't that work? Why would that strategy not work? Yes?
You lose variation, you lose the ability to evolve, and potentially positive, variation.
Right. So you miss out on positive variation, the occasional variant that's actually beneficial in a particular individual or a particular environment. It would make us, when the environment changes, then we would, if we wanted to survive that change, we would have to wait until new mutation occurs. Just in the point out. You'd have to wait until new mutation occurs, and mutation occurs at random. So it could take a long time to get a program that was dealt with the environment. So it benefits the species to have a standing bank of genetic variation in the species. Not every individual and every individual member of that species has that variation, but at least some do. And so when the environment changes, then those individuals, of course, then all of a sudden find themselves sitting at an advantage for natural selection. So it's a good strategy to have that so-called standing variation in the population to deal with changes in the environment. The price you pay, of course, is that some part of the variation is detrimental and those people get sick. So it's a balance between survival of the species and the cost paid by individual members of that species. So it's a clever way to do it that evolution has come up with. But we deal with people who have outlying and detrimental phenotypes. Okay, so what are the types of genetic variation? Well, operationally, a genetic variant is a difference from the reference genome. That's how we define it. And the types, in a general way, single nucleotide variants, SNVs or SNPs, substituting one nucleotide for another. And then insertions or deletions, so-called indels, and by arbitrarily they've been set at anything 49 up to 49 base pairs of size. Elena, I forgot you had a question.
Oh, I had a little example of trying to perfect the variation to whatever standard, and then something happening that ruins everything. They don't do this. Well, with heavy treatments too, but we don't do that. But in agriculture, often growers want to select the perfect variant that grows fast, that grows long, doesn't get that fat quickly, etc. And they did that with bananas at some point. And then, so everybody was going to the same pest. And then some kind of pest killed the banana, this banana variant was susceptible and wiped out all the bananas. And they only, now the bananas we eat now are not the same bananas people ate before.
Yeah, that's a good example, actually.
Isn't the same thing happening now? Like the Cavendish is the common banana that we all consume, but I think there's a fungus that's going around because I think bananas are.
And one thing, when you get a candy and they have banana flavor, it doesn't feel like banana to us. It's because that banana flavor was optimized for the old banana.
Okay.
I know these things.
And then the third category is structural variants. These are just things arbitrarily bigger than 50 base pairs, either deletions or insertions, translocations, duplications, and so forth. And then copy number variants, variations of the number of copies of a segment of DNA, typically arbitrarily set in 1,000 base pairs. And these definitions come from Evan Eichler, and this is a review sort of laying this all out in a straightforward way. But that sort of terminology we use. Now, I put a little asterisk up there on operationally difference from a reference genome. So you should say, well, whose reference genome are we talking about, right? So, for a while, we thought, well, we'll have a reference genome, and then we'll be able to relate everything to that reference genome. But we realized very quickly that was a very short-sighted way to look at it. And so, which reference? And we'll talk a lot about various references in a couple of minutes. But remember that, because some will say, well, this is not in the reference. It doesn't mean it's bad. And it may actually be the most common in some other part of the world. Okay, so if you say that the Genome Project, at least the draft was done in 2003, then in the subsequent 10 years or so, we spent a lot of time beginning to enumerate genetic variation versus that reference. And so we know now, a more nuanced view, we recall that we have 2 copies of our genome in each cell, one from our mother, one from our father. Each genome is about 3 billion base pairs per haploid genome. Encodes about 20,000 protein coding genes. Variation is extensive. Each of us has 4 to 5 million single nucleotide variants as compared to the reference, plus thousands of copy number variants as compared to the reference. And we also know that a single variant can have a profound effect on phenotype. What is your favorite single variant that has a profound effect on phenotype? Sickle cell, yeah. Or achondroplasia. Achondroplasia is a dominant. It's a missense mutation. Almost everybody with achondroplasia has the same missense mutation. And you suddenly go from an average height of 5'8" or 5'10" to 4'6". Yeah.
This is kind of a weird one, but I spent a lot of time in my last position studying codon 12 mutations in KRAS. And we showed in our last paper that KRAS G12D and D mutations are much more oncogenic than KRAS G12R mutations. So just that single, a single amino acid has a big impact. Even though it's changing, what it changes to has a big impact on prognosis for patients.
Right. It's amazing that what you would say a priori is a very subtle difference. Oh, when the difference of protein is 1,000 amino acids long, how can one change of one amino acid have a profound effect on phenotype? But it does. Okay, and that's basically Mendelian disorders. So here's an example of that idea. This is mutations at the FBN1 locus, fibrillin 1 locus. So on the left is an individual with Marfan syndrome. They have tall stature, long limbs, hyperextensible joints, and they have FBN1 loss of function mutations. And on the right is something called the stiff skin syndrome. They have short stature. They have progressive fibrosis, joint contractures, and they have a different kind of FBN1 mutation, missense mutations in a certain domain of FBN1. So a single variant across the entire exome can make a huge difference. Huge. I couldn't believe that when Bart Lois, who worked at that time with Hal, came up with this idea that stiff skin syndrome might be allelic with Marfan syndrome because the phenotypes are so different. But then you say, well, if fibrillin affects height, then maybe you could have certain variants that made people short rather than tall. So it's not such a far-fetched idea, and it turns out to be true. Okay, so those are the sort of formal definitions of kinds of genetic variation, and then obviously in genetics we make huge use of genetic variation. So we use it to give us insight into gene function based on connecting the variant to the phenotypic link, just the kind of stuff we were talking about a moment ago. We use it to explain phenotypes, and we use genetic variation to help us search for variants in genes responsible for rare Mendelian disease. We use genetic variation for molecular diagnosis and risk prediction in the clinic. All of medicine is now using DNA sequencing, and it's essentially a revolution in medical care and diagnosis. It's a substrate, genetic variation is a substrate for genome-wide association studies, which have been so powerful in finding genes that have alleles of modest effect on a particular phenotype. We use it now to calculate polygenic risk scores, which give us some better way of predicting, a more informed way of predicting risk of certain phenotypic features. We use it to understand human ancestry. We already mentioned evolution, evolutionary biology. We use it forensically, extensively, and on and on and on. So the uses of genetic variation and our ability to detect and enumerate genetic variation are just continuing to expand dramatically. And certainly you folks will use it tremendously in your careers. So I just want to go through a couple things, particularly about signal nucleotide variants, because it's a set of terms that people use all the time in and outside of genetics, and I want to make sure that as representatives of genetics, we all use it correctly. So you know that a single nucleotide variant or single nucleotide polymorphism is naturally occurring variation in the DNA sequence at one base pair. So there in this stretch of 10 nucleotides, that A, we're hang on that, paying attention to that A, and on some chromosomes in that position there's an A, and in other chromosomes in that position there's a G. That's a single nucleotide variant. By and large, they're biallelic. That is to say they're binary. There's a few single nucleotide variants where there's two possibilities at a particular locus, but the vast majority of them are binary. That is to say, in this example, we either have an A or a G there. And they occur in a specific position, and attempt to keep track of them all is people use this RS number, so you can go search that RS number, and you'll go exactly to that single nucleotide variant where it is in the genome, what we know about it. And they occur at a specific frequency, which we call the minor allele frequency. Minor has nothing to do with function. It only has to do with the frequency of that variant in the particular population. We call that the minor allele frequency. So we could say if that A was present in 70% of the chromosomes, then we would say the minor allele frequency for the G is 0.3 or 30%. Right? Now, if you went and looked at some other population, it could be different. Right? So the minor allele frequency, when you use it, you have to be aware of what population you're talking about. And that's led people down a wrong path sometimes, because they find a variant that they think is rare in Northern Europeans, and some individuals that are from non-Northern European ancestry, and they find a variant in that population. They say this is a very rare variant because they're looking at Northern Europeans. Maybe this is the disease-producing variant, but it turns out if you look more broadly at that other population, this is quite common. It's clearly not the disease-producing. It's not the variant responsible for the disease. So be careful about that. Okay, so we're all clear on single nucleotide variants? And each of us has 3 to 4 million as compared to the current reference sequence. So let's get back and make one other point, and we touched on this in concepts about the minor allele frequency. So we have some single nucleotide variants that have a relatively high minor allele frequency, like 0.3 or 0.4, and others which are very rare, maybe 0.001 or something like that. So when you see those differences in minor allele frequency, what do you think about? Yes?
That produces like a detrimental phenotype, so the allele isn't carried forward.
So what you're saying is if the minor allele frequency is low, Perhaps it's low, that frequency is low because it's under strong negative selection. And that's certainly one explanation. What's another explanation for exactly that observation?
I guess pollen or like a founder effect.
Not really a founder effect. Founder effect is usually when it's high in a particular population, low in other populations. So it could simply be it's a young one. It just occurred like, 100 generations ago, and so even if it gets transmitted, some variants when they occur don't even get transmitted, so they're in and out of the population in one generation. But if they happen to be in a segment of the chromosome for whatever reason that is fairly passed on fairly successfully, then the minor allele frequency will go up. But if it's relatively recently over evolutionary time, the time is not, the number of generations has not been significant to get the frequency high. So by and large, if you see a high minor allele frequency, that's a variant that's been in the population for a long time. Those are so-called old single-gene type variants, thousands of generations. And if you see a variant that's quite rare, it's either under strong negative selection or it's a relatively new occurrence in the genome. Okay, so just the frequency gives you some idea, but it doesn't distinguish between those two populations. So often people make a leap if they see the frequency of the variant is low, they make the assumption that it's pathological. It doesn't have to be pathological. It could be relatively recent. Okay.
Do you think, with the advent of modern medicine and the prolonging of variation, whether good or bad, we would see an increase over generations in the future?
What do you think?
I don't know. Nuclear war aside.
You said modern medicine, I hope we're not having to deal with nuclear war. But
Well, it's possible. I guess it depends on how detrimental it is. And I'm not able to tell you that.
I think, these frequencies change very slowly over time, evolutionary time. And so there's no question that, I've already said over and over again that as the environment changes, the genome changes the nature of the selection of the genome. And you could think of modern medicine as an environmental variable over evolution, and that will have some changes over long periods of time. And what they will be, I don't know for sure. But it'll take a long time.
I think an interesting observation of that is I used to work in a male fertility lab, and not my professor, but a lot of his colleagues were very apocalyptic about fertility treatments, because they were like, if we prolong, the gene pool of those who normally would not be able to reproduce, then no one will be able to reproduce in 3 generations or something. Maybe I didn't believe that, but it was interesting to have that dialogue in the field where they were like, this is going to be our future, our genetic heritage.
Great. I think many geneticists would say that those changes will be so slow that we'll be long gone. But that's no reason not to think about them, I would say. But I would also say that it's pretty hard to predict what the net effect of selection is going to be in that way.
But also the proportion of the population that is able to access those services.
Yeah, it's
It's
pretty
small, relative to the whole human race,
Yeah.
human population.
Okay.
There's been an increase in C-sections and women needing to have C-sections. Past generations of their mothers having C-sections, so their hips have gotten smaller. And so I don't know if that's also because insurance is pushing for more C-sections, but also more C-sections.
Yeah, I think the access to C-sections across the world is pretty small, or relatively small.
The other option is that mothers die, so.
Yeah.
But you're prolonging their life and their daughters
numerically reduce the negative selection pressure, right? And how fast that would change the MAF, hard to tell. But it would change it. It would increase.
So so I'll give you an example. What about achondroplasia? You heard, let's see, you didn't, I'll get the two classes mixed up. So in SGMD last week, you would have heard, if you were sitting here, you would have heard Julie Woodruff say that women with achondroplasia really essentially cannot have vaginal deliveries. The pelvis is small, the head of the affected fetus is large, and so all women who have achondroplasia, the current recommendation is for them to deliver by cesarean section. But of course, that's a recent, by recent I mean in the last 50 years, 30 years, something like that. And before that, there was plenty of achondroplasia, so why is that?
Older male, fertility. Right?
Well, it's because most patients with achondroplasia have a result of de novo mutation, so the frequency of disease in the population is maintained by de novo mutation at that level. But all of these variables will change in some way or another selection, and selection is going on all the time. And I think a bigger variable, for example, is my favorite one is the development of McDonald's. Right? I can remember when the sign in front of the McDonald's store said over 50,000 hamburgers sold. What does it say now? Over 50 billion hamburgers sold, something like that. Caloric McDonald's and similar kinds of activities has led in the industrialized world to about a 10 or 15% increase in the average caloric intake over the last 30 years. That is an enormous environmental variable. Enormous. And we're paying for it if you just go to the adult clinics and see the consequences of that change in caloric intake against a genome that was never meant to deal with that variable.
Isn't it true that even like some deleterious allele, like if the frequency of that allele was to drop, the rate of mutations hasn't changed in one relation to the next?
As long as that's the paternal age.
Right, because I was wondering if that answers like the whole modern medicine question. Even if you were to somehow artificially like change the allele frequency, the rate of mutation was still, like you said,
Yeah, it'd still be
the
input.
same.
Yeah.
So I wouldn't. That was a false
Yeah.
question.
The way I think of it is that the genome, the environment changes at a much more rapid pace than the genome. And the genome, through natural selection, is always trying to catch up to the environment. So that means that there's always natural selection. And the genome's trying to catch up. And just as it gets, it's like the typical, it gets close enough to grab what it wants, the environment changes, so the race continues in one way or another. And that's the evolution works. Okay. My allele frequency, early origin rate, recent origin or selection. Okay, now this is I want you to make sure you get this right, because if you listen to this, you'll hear it used wrong over and over again. So, what is the formal definition in genetics of a single nucleotide polymorphism? Valeria. You want to consult? Ask one of your colleagues. Yes.
I think that of a sense.
So it's arbitrarily set at a variant that's present in the population at a frequency of 1% or greater. Now, that was a term really that was put in place by Harris and Milwaukee. And they wanted to study variation that was extant in the population, not new mutation variation. So they said, we're gonna forget about really rare variants because they might be a mutation. And we're only gonna take variants that reach a certain frequency that's much greater than could be accounted for by just new mutation in that gene. Okay. And they decided they'd be at least 2 or 3 orders of magnitude away from that, so that they picked a very high frequency, 1%. Okay. And they said anything at that frequency or greater, we're going to call it as polymorphic in the population. So the definition is one based on frequency, and it has nothing to do with function. Nothing to do with function. Okay. Now, people thinking in a shorthanded way say, well, if it's common, it must be benign. And I think you all know instances where that's not the case, right? But it is a term of frequency, not a term of function. So if that's the case, what do these comments mean? A rare SNP. You have all heard people say this is a rare SNP. Well, I think when I hear that, I think, what the hell do they mean? Right? Okay. A monomorphic SNP. What is a monomorphic SNP? That's a
Wait, I don't get it.
That's a SNP. It's not a SNP, right? Yeah. Sometimes people do use it correctly, and they say this single nucleotide variant in this population is monomorphic in another population. Okay.
So it's not a SNP in another population, is basically what that means?
Right.
Yeah.
Oh, okay.
A lot of this, I'm going to try to convince you that a lot of this goes away if you use single nucleotide variant rather than single nucleotide polymorphism. It's just a SNP. Now, what does that mean? I would say, well, a single nucleotide variant, I wouldn't belittle it as saying it's just a single nucleotide variant, because it's a change in our genome. It may or may not be under selection. We may be able to use it to tell us issues of evolutionary history of that region of the genome, or to find genes or whatever. So I don't belittle single nucleotide variants. I would say that when you say it's just a SNP, it's a sort of a pejorative way of saying that it doesn't count for anything. And in fact, you can use it in lots of different ways. So I don't like that. You could just say it's a single nucleotide variant, period. So the key point here is that not all relatively common variants are neutral. So a lot of probably sickle cell disease or other things, right? And that polymorphism is a term of frequency, not a term of function. You will hear particularly non-geneticists use that. The thing is, it's a great example of the power of an acronym. If you come up with a catchy acronym, it sticks, even if people don't use it historically. Okay? Use single nucleotide variant. Avoid sloppy language and sloppy language. Okay, now we're going to talk about some functional consequences and other aspects of variation. These are some useful databases. Slides in this color scheme I've taken from a lecture given by Stelios and Tonarakis at the Bar Harbor Short Course, which we'll go to in July. You'll hear him lecture in July. So these are his slides when you see this font and this color. And they're a little out of date, but you can go to these websites and update the numbers if you want. And so there's OMEN, you all should know how to use OMEN. There's the Human Genome Mutation Database, HGMD, which is maintained by the group at Wales. And ClinVar, which is a relatively recent one, but is very well annotated. Very well annotated. So all of these numbers have been increased since the time this version was in 2021. So now I want to talk, since we said that, we alluded to the fact that we have to pay attention to which references, which reference genomes we're talking about. Is that cloud true? No.
No.
11, 18.
Okay, so the reference genome was generated by the Human Genome Project, which was completed in the mid-1980s. It was debated and argued. People thought it was a bad idea. Many respectable scientists thought it was a bad idea. It was a committee put together by the National Academy of Sciences of experts was tasked with debating and thinking about the idea, and ultimately that was called the Alberts Committee, chaired by Bruce Alberts, included from here Victor McCusick and Dan Nathans. And ultimately that committee, which I think was skeptical when it was convened, came to the conclusion that this would be a good idea. And that their deliberations lasted about 18 months. So based on the support of the Alberts Committee, it was elected to fund the Human Genome Project, and it was started on October 1st, 1990, under the direction of none other than your friend and colleague, Dr. James Watson, who lasted about 18 months in that position before he burned all his bridges in Washington and was taken over by Francis Collins. So the initial efforts, and this is all from suggestions from the Alberts Committee, were to focus on improving technology, sequencing technology, technology to develop maps so we sort of had a sense for where we're going in the genome, and that these strategies would be tested and sharpened by using model organisms that had smaller genomes and were less expensive and less complicated to deal with. So it started on October 1st, 1990, and as it moved forward, it eventually became a race between two groups, the so-called public sequencing team, which is headed up by Francis, and centered at NHGRI and NIH, but included contributions from scientists all over the world. And then the private sector, which was Celera Genomics. Celera means fast. And that was headed up by Craig Venter. That's Craig and Francis in that picture. The public project was decided to go on a so-called bacterial artificial chromosome, or back-based strategy, and so they took individual genomes, broke them up into large fragments, and made that could be cloned in a certain kind of bacteria to make a bacterial artificial chromosome. The value of that is it's very stable, and the insert pieces were on the order of 100 to 150 kb, so they're 100 to 150 kb chunks of genome. The various sequencing centers, a set of 4 or 5 sequencing centers around the country, they would apply, they would get a back, and they would then break that back, insert up, and sequence it over and over again, and then put it back together again, and then they would return that sequence from that back to the database. And the backs all had what are called back-end sequences, which were the ends of the insert, and you could use that to overlap the different backs using the back-end sequences. Okay? And the private project, the idea was to break the entire genome into small fragments, sequence everything over and over and over again, and then put all the sequences in a computer and push the button and ask the computer to align them, overlaps, and spit out the human genome at the other end. So 2 completely different strategies, right? Venter argued that his method, the private group's Celera group, was better. He had to put together a huge computing resource, which he did. And the other thing that was different was, it's very difficult in the private project, you couldn't tell where you stood because you had to put everything together and then push the button and get the answer. Whereas the public project, you went back by back, and you could say, well, you've got this many kb on chromosome, whatever, this many kb on that. And the public sequence group made it clear that when a center completed a back sequence, they had to put it into the publicly accessible database within 24 hours. So each of these big centers downloaded their back sequences, complete back sequences, every 24 hours. So you could begin to go to the database and use pieces of the part of the genome that you were interested in was very whereas you couldn't see the private one because it hadn't been assembled yet. Now, of course, that was fine with Craig because he wanted to patent the genome sequence and use it for commercial purposes. Magically, they both finished at the same time. And there was a meeting at the Clinton White House. This was the year 2000, in which they had both just a few weeks before announced the completion of the genome. That's what this headline on the New York Times is about. And they were smiling. They were there at the Clinton White House celebration. Now, subsequently, people, including Steve Salzberg from here, have taken a careful look at the sequences in the public genome. And it turned out, you would like to say, well, okay, I want to capture variations, so I should get my back libraries from a number of different people. Right? And at the time, there was only one lab, which was headed up by a guy named Peter Dijon at the Cancer Center at the University of Buffalo in upstate New York. There's a football team up there. But later, he was very good at making back libraries. His institute was Roswell Park, so that's RPC, Roswell Park Cancer Institute. And nowadays we're not allowed to say, go to people in our lab and say, give me a sample, give me a But in those days you could do that. Okay, so what Peter Dijon did, he had a grant from NIH to make these back libraries. He just went to his lab and up and down the hallway and made these back libraries. And it turned out he made a really good one on RPCI number 11. And that was a technician in his lab. And if you look at the original reference genome, this one person contributed more than 50% of that genome. And that one person was an African American, a mix, a pretty even mix. I don't know the extent of the mix. And so I happened to be on the advisory part of the advisory group, and we had a meeting, and Francis got up and he said, "We have a problem." He said, "We realized that one individual, because his DNA made a very good back library, his DNA is being used disproportionately. And there was no consent signed. He just was in the lab and said, okay, I want some blood from everybody. He didn't sign a consent. So Francis said, we have two options. One, at this point we were 11% into sequencing the human genome. He said, we can throw away all the sequence that we've got, consent the people, and start over again. Or we can reconsent them right now, but retain the sequence that we have. So which choice do you think the committee decided upon? You're absolutely right. As we were in a race with Celera genomics. So, in any event, the final product, as shown by the diagram on the left, was disproportionately from one individual, who ultimately was consented, but not until the process was underway. And you can see the makeup of the others. Now, Celera had the same problem. This is not so well known. But when the Celera genomics genome was put together and we got more genomic information, it turned out there were a number of sequences in the initial Celera version of the whole genome that came from a dog. And if you had visited Craig Venter's office at the time, he had a standard black poodle named, I forget what his name was, but that poodle was everywhere. He lived in Craig's office. I can remember riding the elevators and the poodle was going up and down the elevators and everything. And I'm pretty sure that poodle, the DNA in the original Celera sequence came from that poodle. But that was cleaned up. So So one of the things that was done as soon as the references the draft reference was done, that New York Times thing, was recognition that now what we needed to do was to get genomes from lots of different people. In fact, if you look on the 5th floor of MRB, there's a poster there of a symposium that was put in place that was to celebrate the opening of the Institute of Genetic Medicine, the predecessor for our department. And one of the speakers at that meeting was Francis Collins. And I think this was about 2001 or something like that when the symposium occurred. And Francis gave his talk about nearly completing the draft of the human genome. And someone raised their hand and said, well, now what you need to know, that's just the first step in this. What you need to know is the extent of genetic variation across the world. Now, who do you think raised, yes, ask that question? Barton Childs. So very shortly after the completion of the draft, then a variety of projects, initially what was called the HAT map, and then 1,000 genomes, but now a whole bunch of projects as shown in blue down below this. And this slide is out of date now, but getting sequenced. So we've gone from very few whole genome sequences to billions. And you're the beneficiary of that. You can dial it up. And there are lots of databases, and you should know those databases. So this is a screenshot from the NOMAD database, and you should know what's in there and how you can use it. And there's lots of information, including all of the, NOMAD is a very clever effort to take people that were ostensibly healthy. Their criteria were they were adults, so they didn't take anybody who was in the pediatric age range, and they took people, they tried and are continuing to try to diversify the ancestral origins of people that contribute to the data, and by the fact that they live long enough to be an adult, they're assumed to be more or less healthy. And but I'm sure, how many of you have used NOMAD? Have you all looked at NOMAD? You should all look at NOMAD. Just pick your favorite gene, type in the browser NOMAD, and it'll come right up, and then it has the first box, put your favorite gene in there, and you'll get a page like this. So these blue humps are this is for a gene called ornithine aminotransferase. These blue humps are the exons, and this exonic sequence, and the green are whole genome sequence, so it includes the introns between the exons. And if you look now, this slide was made a couple years ago. If you look now, most genes, the green and the blue are the same height. That is to say, the reads across that region are essentially the same, and that reflects a lot of whole genome data going into the NOMAD database. And if you scroll down here, you'll come to one end or the other of your gene, your favorite gene, and you'll see, and you just keep scrolling down, and you're going along the length of that gene, and you'll see every variant that's been reported in the NOMAD database that's in that gene. So you can say, I found this variant, has it ever been seen before? And you can go into NOMAD and you say, well, it's been described 6 times in NOMAD, and 3 of those times were from Northern European people, and 2 of them were for East Asian people, and so forth and so on. So you can get the frequency and the location of the variants, and a judgment of classified as pathological, likely pathological, neutral, and so forth. And you see this table of constraints, and basically I can't explain it in great detail right now, except to say that by looking at the sequence, they can say how many synonymous variants are you expecting to see in a stretch of DNA of this length, and then how many were actually observed. And if the observed are far different from the expected, then the gene is under some sort of evolutionary constraint, or changes are under some sort of evolutionary constraint. And so you can see there shouldn't be much evidence or constraint for the synonymous, but perhaps for the missense, you would see it. So in this case, there are 200, for missense, there are 242 expected and 204 observed. And that's a z-score of 0.88. And then another category predicted loss of function. So there's a lot of information in there. Learn how to use it. And the best way to use it is to pick some genomes in there and play around with it, okay? Now, most of the data initially in Nomad was exome, now more and more genomes are in there, but a different program sponsored by Heart, Lung, and Blood called the TopMed program wrote in a lot of whole genome sequence. At the time of this slide, which is 2021, there are 53,831 whole genomes. The advantage of the TopMed is that they sequence a bunch of longitudinal studies supported, a bunch of individuals in a bunch of longitudinal studies supported by Heart, Lung, and Blood. And those studies very often have a lot of phenotypic information. They have routine chemistries, they have proteomics, they have metabolomics, a lot of information that is not attached to the data in Nomad. So if you want to connect variation to some of those phenotypes, a good place to look is in TopMed. And this will list all the variants and so forth, and the front end of the browser looks very similar to the Nomad depiction. But go to that TopMed Bravo website and take the same gene and look and see what you can find out about it in Bravo. Okay, so those are just building the reference genome by sequencing more and more people from different parts of the world. Now, technologies come along, and so we can improve the reference not only by sequencing more broadly, and for example, Amber Wacom has been the leader advocating for sequencing at least 3 million African genomes. There will come in a minute to the heterogeneity in the genetic heterogeneity of people in Africa, who live in Africa, and different environmental conditions there. So there's a lot of diversity to examine in Africa, individuals who live in Africa. And then this project, which you've heard about, I'm sure, the telomere to telomere project, in which we took advantage of long-read sequencing, and we took advantage of sequencing a hydrotypical mole, which is homozygous or hemizygous for every for the genome in there. So there's no heterozygosity; it's all homozygous. So that gives you the haplotypes unambiguously across the chromosomes. So that was a big breakthrough. And then the one that's going on right now, the so-called pan genomes, is sequencing healthy individuals, a large number of healthy individuals from different populations, and then figuring out a way to display these results in a way that we can interpret them. So this is an issue of science where the telomere project was, the final project was reported, and there's a fair contribution left here, Mike Chaffee's lab out of my efforts, and what's his name? Lecture.
Liliana?
Liliana.
No, sorry.
Liliana. Liliana was
Yeah.
very involved in this Celera genomics effort, did a lot of the bioinformatics behind that.
Yeah.
And then Rajiv McCoy is going to talk to, will talk to you. He was a big player in the T2T as well as Mike Chaffee. So we look forward to those lectures. And there's effort, not only this effort includes not only single nucleotide variants, but big chunks of chromosomes variants in structural variants that are detected and reliably by long read sequencing. And then this pan-genome project in which healthy individuals from different populations put it together, and you'll see copy number variants, and see assigning an unambiguous number to a particular base in the genome is very difficult, because what's that position 1,573,492 at my chromosome might not correspond to the exact same place in somebody else's chromosome. So having to figure that out and to describe reference sequences that allow for length sequences is in progress right now. So that's going on quite intensively right now. And then one neat thing that's also going on is the ability to sequence ancient DNA and to look in more carefully at this observation that each of us has some Neanderthal DNA in our genome on the order of 1 to 2 to 3%. So this paper just came out, and they looked at, they were able to sequence a larger number of Neanderthals and a larger number of ancient human species. The bottom line was shown down here that they argued that this integration of Neanderthal DNA into Homo sapiens DNA took place mainly over a 7,000 year period in Europe. So when the people who came out of Africa occupied the same territory as the Neanderthals who had already established themselves in Europe. So this intervening was about 7,000, occurred over 7,000 years, and there was a lot of selection once those two genomes were scrambled up. So there was a lot of variants that were purged out, and then some variants that were selected for. And now we have this sort of fraction of Neanderthal DNA that's sort of pretty much in all of us, except individuals of African descent, because they didn't migrate out. So they didn't come into the Neanderthals, except to the extent that there was some back travel. So we're learning right now a lot about the evolution of our genome, and it's very interesting to understand, well, what there was a variant in Neanderthal and it seems to be retained because it was beneficial. What was the nature of the selection that was going on? And there are some variants, of course, that you recall that appear to provide resistance and or sensitivity to COVID-19 that we tied into our Neanderthal history. So if you're interested, read that paper. And all of a sudden, this is not working. What time is it?
11:40.
I can move slides for you.
Okay, so I'm supposed to finish
12.
at 12, okay, all right, we do it, Cody, let me see now, let's do. Wait,
Oh, yeah.
okay, so we talked a lot about the nature of the variation, the extent of variation in our reference genomes. Let me say a word about the origins of genetic variation. So next slide. So this is a slide from Stelios, and we think that the mutation rate is about 1 times 10 to the minus 8 per nucleotide per generation. The maternal gamete mutation rate remains stable. The paternal gamete mutation rate doubles every 16.5 years of paternal age. Or you can think of it as approximately 1 to 3 additional new paternal mutations per year of paternal age. So the average newborn human has 50 to 100 new mutations. And about one of those, or here 0.86, change in amino acid, and 5% of the nonsense. Now, most of them occur in non-exonic parts of the DNA, so that's why you go from roughly 100 down to 1. Everybody with me there? Okay, next slide. And here's a breakdown of the kinds of variants that mutational process produces, and you'll see that the biggest category are missense mutations, and then followed by small deletions, and then nonsense mutations, splicing, and so forth and so on. And this is based on over 300,000 pathological mutations that were known in 2021. Next slide. And the most common is changes from so-called CG changes, because cytosine in the DNA undergoes spontaneous methylation to make 5-methylcytosine, and that undergoes deamination to make thymine. So amino acids whose codon includes a CG are the ones that are most involved in missense mutations, which is largely RG. Next slide. And of course, I think you all know the sequencing strategy that we detect so-called de novo mutations is this trio sequencing, where you sequence both parents and the offspring and look for variants in the offspring that are not present in either of the parents. Okay? Next. And depending on the paternal age, as I say here, I've already said about 100 de novo mutations per generation. You expect 1 to 2 to be in the exome. And these de novo mutations contribute to many phenotypes, especially those in which the reproductive fitness of the affected individual is low. Does anybody recall why that's so? Anybody want to have a crack at that?
Can you ask the question again?
Yeah. Why would you expect that de novo mutations play a larger role in producing phenotypes in which the reproductive fitness is low?
Because they're de novo, it's not like something that would have affected either of the parents, potentially. Like if it's a de novo, it's not something that would have affected the reproductive fitness of the father.
I think I'm having trouble hearing you, but I think you basically are saying the right thing, which is that if it were not for de novo mutations, and since these individuals have a low reproductive fitness, the phenotype would disappear, right? Because individuals of those variants would not transmit them. But these phenotypes are not disappearing. For example, schizophrenia is 1% of the general population, and a large fraction of, and they're, particularly males with schizophrenia have a very low reproductive fitness. So their genetic variation that's producing schizophrenia in them is going out of the population. But the disease frequency is remaining stable, so there must be an appropriate number of de novo mutations replacing those each generation. Okay. The observation, of course, was that advanced paternal age is associated with increased rates of de novo mutation. And this paper is a classic one, and we'll make sure that it's in your in Canvas. But this is from Decode Genetics back in 2012, and they were interested in schizophrenia. So they did trio sequencing, which was not as widely done back then, and they asked, well, what was the relationship of the number of de novo variants in the child compared to paternal age. And you can see there's pretty much a linear relationship. So similar data were available from an Israeli population in which they calculated that 90% of the genetic contribution to schizophrenia was due to de novo mutations of young paternal age. So it's a big effect. And it has to do with gametogenesis. Next slide. And so here's males on the left, females on the right, and the number of replications of the germ cells. So this is just a pre-germ cells with replications making the embryo. Then after the gonads develop, females produce eggs, and then remember, the most eggs that a female individual has is when she's about 5 or 6 months in utero, and then that number goes down from that point as various eggs die off, oocytes die off. But in males, the process continues. So in post-puberty, in males, there are about 24 replications, spermatogonia replications per year. So a large number of replications, and each replication, of course, has that mutation. So advanced maternal age is associated with mutation. So people say, well, de novo mutations aren't inherited. They are, in fact, inherited, right? They're inherited from the person who had the de novo mutation. But that mutation occurs in the germline of that person, not in their sons, so they don't manifest the phenotype. So they're transmitting, it's a transmitted mutation, it's transmitted to the person that has the phenotype. And there's this interesting paper, I'm not gonna go into it, about looking about meiotic DNA breaks being involved in increasing the mutation rate in some clusters of mutations around regions of the genome where there are meiotic breaks in the breaks in the DNA during the process of meiosis. So I urge you to take a look at that paper. Next slide. We already went through this slide about the uses. Next slide. And just in closing, I want to just say a few comments about sort of the population genetics, using genetic variation to understand population genetics. So, as I said, Homo sapiens arose in Africa, and about roughly 100,000 years ago, a population came out of West Africa into the Middle East and ultimately into Europe and then to the east over to Asia. Africa itself is Africa itself is very heterogeneous, and there are at least 14 evidence for at least 14 ancestral populations in Africa. So when someone says this person is African, that doesn't really tell you that much about their ancestry because there's lots of different populations in Africa. And these data, these pie charts show heterogeneity of the genomic variation in individuals in different parts of Africa. It largely follows what are called ethno-linguistic groups. These are people in whom you can tell from their language they've been separated from one another for a long time. There was a human geneticist named Luca Cavalli-Sforza, who's a population geneticist, born who worked in the pre-genomic era, and he used the language to help him distinguish into the differences of human populations. And by and large, his predictions based on language are right on, supported by genomic data. And so, current leader in this area, I would say, is Sarah Tishkov, who's at Penn, but you'll hear her talk at Harvard. So lots of heterogeneity, both in terms of genetics and environment in Africa, which we can assess now because we can do this genomic sequencing. Next slide. And the same kind of thing you can see in Europe, and there's a person named John November. I can't remember his
Yes.
said his first name. Has sort of pushed this and used it to point out that you can look at the pattern of variance in the genome sequence of a particular individual whose ancestors come from Northern Europe. And you can tell exactly, almost within 100 miles, of where their ancestors lived in Europe. And this is using principal component analysis laid out on the map of Europe and looking at the pattern of genetic variance. And you can see a cluster in the Sardinia, Spain, Portugal, and so forth and so on. So you can use this genetic variation to really determine where an individual's ancestors were, and also the origin of a particular variant that might be associated with disease. And you can do the same kind of analysis in other parts of the world, including Africa, shown in the lower right. Next slide. Now, just a word about pathologic genetic variation. Next slide. So we keep track of it in OMIM, which is kept by Ada Hamish and her team over on the 10th floor of Blaylock. These are the current data. There's about 4,956 genes that are associated, that have variation that are associated with phenotypes. It's about 25% of the total genes in our genome. Explains about 7,581 Mendelian phenotypes, and there are still 1,000 unexplained ones, and new ones come in every year. So we're continuing to expand the number of genes which have variants of major effect and produce Mendelian phenotypes.
How are variants added to
How are they added to OMIM? By hand. So typically, OMIM, the raw data for OMIM is the published literature. So they scan the published literature, and if they look at paper and it looks like a new disease gene or a new variant of significance, then it gets into OMIM. And in the past, students have participated in the OMIM writing team. I don't know if they're still doing that. I think probably they are. No, I don't know if they still are or not. But if that's of interest to you, you can come see me, or you can make an appointment to see Otto and talk to her about that. It's a characteristic style of writing, so it's very formulaic. But in any event, the key thing is the data they use is the published literature, so they don't go find ClinVar, they don't go look at NOMAD. They may look at NOMAD to decide if a variant really is responsible for a phenotype, but they don't just say, okay, we're going to port over all the variants in NOMAD and stick it into it. And OMIM makes no effort to list all of the variants. They really, their remit is to connect genes to phenotypes. They'll have the salient variants or important variants, but not all variants. Next slide. Now, one benefit of that, of course, is treatment, and so the amazing progress with gene editing and so forth is really changing our ability to treat genetic disease, and in your lifetime, that will be a major driver for the way we think about genetic variation and what we do about it. Next slide. And here's a woman who, one of the first patients who had ex vivo gene editing for sickle cell disease, and she's, as far as I know, without any sickle crises since this work was done. So she's a real pioneer, and very exciting. Many challenges, however. A big one is delivery to the right tissue for each disease, and also allele heterogeneity. For sickle cell, of course, you just have to deal with one variant. But if you have a gene which has lots of disease variants, then we haven't really figured out how to make that pipeline really efficient and effective. Next slide. Here's a boy who went from not hearing to hearing by gene therapy, which is really exciting. Next slide. Now, in the last couple minutes, I want to talk about diversity and how we can use diversity, but also some problems related to diversity. So, a number of groups have realized that if you go to a population which has not been studied, and you study your favorite phenotype in that population, you may find different genes and different variants that produce that phenotype in that population. So this paper, which is published by Ambrose just recently, 2005, did exactly that. They went to Mali, where there was a group that was collecting families that had congenital hearing loss. And they collected those families and did whole exome sequencing to see if they could find a genetic variation that was responsible for that hearing loss. And the background info, you're ahead of it, go back. No, back one, right, okay. So the background info that childhood hearing loss is about 50% genetic. Hearing, that HI is hearing impairment. And we now know of more than 120 genes which produce hearing impairment, and which mutations can produce hearing impairment. If you look at the complexity of the ear, you would guess that maybe there's another 60 genes to go. So how can you find those new disease genes that have yet been found? So what they did here was they took 24 families that they identified that had multiple affected individuals, suggesting it was genetic in origin, subjected them to whole exome sequence, and they were able to find the gene and variant responsible in 18 of the 24 families. So they had about a 75% success rate.
And what they found was 12 genes are already known to produce hearing impairment. They found new alleles, but they didn't find new genes. And one gene that had not previously been associated with hearing impairment up until now. That's a pretty good hit rate, and it's a nice strategy to find new disease genes and new variants that produce hearing loss. However, next slide, or hit it again there. So it's a powerful strategy for disease gene discovery, but does it benefit the subjects? So that's a question that, as human geneticists, we've had to make sure that we pay attention to. Because if you look back in the history of these kinds of studies, there have been studies in which this kind of study was done, and the information, while it benefited genetics science, it didn't benefit the people who gave the DNA. So I'm not saying this study was very thoughtfully done, but previous examples of this approach had that notion, or that problem. Next slide. So that came to be called helicopter genetics. So the idea that you helicopter into some remote population, you grab the DNA you need, and they never hear about it after that. And I think we would all agree that that's ethically improper, and it's just not fair, and it shouldn't be done. So this term has been used to describe studies in which the people who contributed the DNA have no access to any medical benefits that come after based on that knowledge. No access to any monetary benefits if this information is somehow hooked up to big pharma or something like that. And the population has the potential for stigmatization by the results. So in other words, they can say, well, this population has more diabetes, or this population has more alcohol as a genetic basis. So what we've learned, some of the stuff that we've learned, and this is just my quick enumeration of it, is that we do these kinds of studies can be very valuable. But if you're going to think about doing it, the first thing you need to do is become culturally educated as to the population, and that takes work and cooperation. And very often involves what's called community consultation, where you go meet people from the community and find out what's important for them. What we think is important might not be important for them, and something that we don't give a second thought to may be very important for them. And so you have to find out those values. Sometimes you actually have to consent the community, because the community is, in some populations, the elders of the community control the community, and they have to say, even though they don't have the DNA, they're not affected. So a lot of pre-participation work and become culturally aware and do individual counseling, not only in the population, but also to the participants. And then, very often, making formal contractual agreements that ensure that the subject will have access to any medical and or monetary benefits from the data, and that the participants control any subsequent uses of the sequence information. So people may say, okay, we'll give you some money for this, or we'll make sure you have medical benefits, but then we're going to sell your DNA to somebody else, and then you get to do something else that the participants had no knowledge of and no control over. It's not right. Okay. And there are examples of studies that were done just exactly that way and had exactly all of those negative consequences for the study participants. Next slide. So just recently, this model has been sort of put forward, and there's a company called Variant Bio, and they're based in Seattle, and these are some people from Variant Bio talking to participants who happen to be individuals of Maori ancestry in the South Pacific. And they had some phenotypes that people in this company, in this little biotech company, thought might be beneficial to understand. And they made a contract, after a lot of interaction, they made a contract that any medicine that is invented based on these data will be made available to these people. Any profits that will come out of these studies will be, they will get a share, the people will get a share of the profits. There'll be no patenting of their genetic information by the company. And there'll be no additional uses of the sequence information beyond what was contracted at the original agreement. Now, this company just got bought out by Novo Nordisk, so we'll see how good their contract is. But it's at least heading in the right direction for how these studies are done. I think we would all agree that as geneticists, we should be involved in this, we should be in the lead in this, we should understand what the genetic information means for people whose genes are represented, and how this genetic information can be used, and make sure that these sort of little guidelines that I've listed in the last couple of slides are part of any such study going forward. And if done in the right way, the results can be valuable in terms of advancing science and advancing medicine, and maybe even in terms of financially. But if done in the wrong way, they're terrible, and we'll lose trust. And so it's our responsibility to make sure these studies are done properly. Your responsibility is going forward. Okay? And you'll hear more about that in this course. Next slide. Next slide. I have two last slides I want to show you. I'll put this in the references, but this is a picture of Maynard Olson, who's retired now, but he was one of the thought leaders of the Human Genome Project. He wrote this very interesting article, The Human Genome Project, A Player's Perspective, and he was a player. And so he's the guy who figured out how to make back on him, basically. But he's a very thoughtful person, and I think you'll find this interesting to read. Next slide. And last, the irony of having the discussion, particularly the last part of this discussion on today, when our government has just eliminated all of the efforts for DE&I activity, is quite the irony is quite great, I would say. I think it's not for me to transmit my own personal ideas about this. Each of you can make your own personal ideas. But it is for me to make sure that studies done on individuals of diverse populations are done properly. For me, number one. And number two, I think it goes without saying, if you think about everything we've covered in this lecture, that diversity amongst our species is fascinating and highly variable and biologically expected. So diversity is not something that is bad or is to be avoided. Put your head in the sand and say there's no diversity. You're missing one of the wonderful aspects of life, basically. So it's hard to believe that this is going on. I'll leave it up to you to make your own decisions about it. But it is our responsibility to educate people about the value of diversity, the normal expectations for diversity, and to eliminate sort of misunderstandings and erroneous conceptions about diversity. Thank you very much.